Okay, hi everyone. I'll just get started now. Thanks for coming to my talk. My name is Christina Steinecke. I'm a graduate student in Robin Hopkins Lab at Harvard University. And today I'm going to talk about a project looking at ecological divergence and plasticity in flux annuals. So by this stage of the conference, I'm sure all of you are aware about the vast diversity of phenotypic variation in plants, but if you'll allow me to divulge a little bit. When a seedling emerges from its seed, it's essentially stuck in the place where it grows for its entire life. And this immobility really forces this plant to be dynamic in its phenotype in order to survive, to reproduce, and to grow. And this dynamic change can occur across micro and macro evolutionary timescales. But if you're interested in plasticity, like we all are, you'll think of it more as changes within a single generation. So phenotypic plasticity is a strategy that allows organisms or genotypes to respond adaptively or non-adaptively to varying environments. It's most often thought of at the individual level, where a single genotype can use environmental cues to express a suitable phenotype, but we know that it can also operate on the level of populations, where multiple genotypes of the same species occur in similar conditions, as well as between species or among species. So what inhibits or allows the evolution of plasticity? Although the optimal response to a set of environmental challenges is perfect plasticity, where a genotype gains perfect information on its current environment and uses every mechanism to produce the right phenotype response at all points of its development. This perfect plasticity rarely occurs, which suggests that there are some sort of constraints on the evolution of plasticity. And these manifest as inherent costs in the form of energetic trade-offs or genetic limitations, where standing genetic variation is just insufficient to produce a plastic response. So for this talk, I'm just going to focus on genetic limitations and show you a really cool example looking at plasticity across a group of closely related species rather than within an individual or a population. So, that was an animation I missed. So for this talk, let me first introduce you to Phlox cuspidata, also called the pointed phlox, Phlox drumundii or Drummond's phlox, and Phlox rameriana, also called the golden-eye phlox, who I think is the prettiest. So these three little cuties make up a very closely related clade of annual phlox that are broadly distributed across Texas and whose habitats we know surprisingly little about in the literature. But what we do know, especially now that we've all attended Felix Wu's talk on the genomic landscape of these three, we know a lot about their genetics and the standing genetic variation. So relative to Phlox drumundii, shown on the bottom here circled with blue, Phlox cuspidata and rameriana have much lower effective population sizes and relatively lower nucleotide diversity, estimated with rad loci. And what's even cooler is that we suspect that their low genetic variation arises from completely different evolutionary histories. For Phlox cuspidata, we think it's likely because of a loss of self-incompatibility mechanisms or an ability to self-fertilize. And in Phlox rameriana, we suspect that it's due to it being restricted to limestone outcrops and being an ecological specialist. So this begs the question, given that Phlox drumundii has higher standing genetic variation, is it more plastic? So to start answering this question, we wanted to get an idea of the types of environmental variables that we could potentially manipulate, and what about their environments were interesting or similar across species. So we did some maxent distribution modeling using a combination of GBIF and lab-collected data, iNaturalist points, basically, as well as WorldClim variables, and found that there's a pretty strong overlap. My points didn't show up. That's annoying. There's a pretty strong overlap in niche preference. So imagine this is like one big circle of points across these species. And when we looked at the contribution that each climate variable had, we found that the top 4 variables, which cumulatively explain about 90% of presence related to each of these species, is related to precipitation. So this shows that the flock's niche is determined mostly by water. But this is at a pretty large geographic scale, and so we wanted to see if this can be recapitulated on the ground in actual soils. So we went out to Texas and sampled soils from across each of their ranges and found that generally their soils are quite different, and that again, most of this difference is driven by water availability. So now I've shown you in 2 different ways that water availability is important for these species. So with all of this environmental information, we hypothesized that Phlox drumondii is more plastic given that it has higher genetic variation relative to its relatives. So to answer this question now, we sampled seed from across the ranges of each of the species and grew them up in the greenhouse. We grew 2 to 16 siblings from each family of each species and split them equally into either a wet or a dry treatment where soil moisture was manipulated. They were able to grow in these wet and dry conditions for about 2 months until they grew to maturity, at which point we measured a bunch of floral and vegetative traits to estimate the plasticity response of each of them. So here I have a reaction norm with treatment on the x-axis and an example trait, dry biomass, on the y. And what you can see is that shown in blue, Phlox drumondii's phenotypic response was really drastically different in wet and dry conditions, meaning that its slope was higher, as shown as the M value. But this was true for each of the other traits? Plotted here, I have each trait that we measured on the x and the value of the slope on the y. So here where a high slope value indicates a huge jump from dry to wet conditions, indicating a pretty plastic response. So if we break this figure down, when you look just at the floral traits, you see that Phlox drumondii does not have higher slopes than the other annuals, and therefore we can say that it's not more plastic in floral traits. But when you look at vegetative traits, you see super clearly that drumondii has a much steeper and stronger response to treatment. So if we do kind of a hand-wavy job at consolidating these traits, we might conclude that Phlox drumondii is vegetatively very plastic, and florally it may be constrained by genetic architecture or development or something like that. So we know that Phlox drumondii is pretty plastic according to individual traits, but is it plastic overall relative to other Phlox annuals? Or in other words, is it more plastic in its multivariate phenotype? So the last thing we did was a dimensionality reduction where this shows firstly that these species cluster really nicely according to just general phenotype regardless of treatment. But when you delineate these points by wet and dry treatment, you see that while each species seems to be split pretty evenly, Phlox drumondii's individuals in wet and dry conditions form 2 very disparate clusters, indicating a really strong divergence between wet and dry conditions. Therefore, we confidently share that Phlox drumondii is more plastic generally than the other annual phloxes. So, to summarize and conclude, today I've shown you a story and described how the distributions of annual phlox overlap and have a strong reliance on water. The soil moisture differs significantly among these annual phlox, and that Phlox drumondii is the most plastic of the Texas annuals. And overall, I've demonstrated that genetic variation has a really strong influence on the capacity to plastically respond to the environment, and that this can be true regardless of evolutionary history. So thank you for listening. Thanks to my lab and all the funding sources, and I'll take any questions. I would argue that it is potentially adaptive, but that's something that I'm actually really interested in figuring out, potentially by doing a reciprocal transplant of Drummondii or whichever species from either side of its range, because we know that there's variation in soil moisture along the range. But yeah, I would love to know. Yeah.
So soil moisture is lowest in that central area. Is the variation in moisture also largest there as well? So both droughts and wet areas?
Yes, it is. Yeah. And so that's why we wanted to sample from across the entire range of each species to make sure that we are capturing that variation. Okay, thank you.
Hi, everybody. My name is Baxter Worthing. I'm a PhD student at the University of Vermont. And today I'm going to be talking about the genetic architecture of bud flush timing in poplar trees. For those of you who aren't tree biologists or don't look at trees throughout the year, just a reminder, trees generally go dormant during the winter in temperate parts of the world. That means they stop vegetative growth and lose their leaves. And then in the spring, once winter is over, they undergo this process called bud flush, which is the focus of my talk. So I'll mention that bud flush looks like, can you see my cursor? Bud flush looks like that on my title slide. It's the moment when a once dormant bud on a tree branch opens and releases new leaf growth. Now, we think of this, the timing of this bud flush process as being incredibly important to the tree's health. And it's essentially an adaptive trade-off between the need to maximize the length of the growing season, while also avoiding the risk of flushing too early and exposing oneself to the danger of late winter, early spring frost. And so this individual here has made that mistake and accidentally flushed its buds too early and then got hit by a return to winter temperatures. And what ends up happening is a few weeks later you look like this. And so frost damage can be really detrimental to a tree. You can lose an entire branch, and that's just a bunch of wasted energy. So in order to guard against this, poplars and other trees have evolved basically two physiological locks that need to be unlocked before they exit their dormancy. So the first we call endodormancy. That is released or broken by the accumulation of cold temperature. So it's essentially a check to make sure that winter is over. And then that second physiological lock that needs to be unlocked we call ecodormancy. And so I did start early. We call it ecodormancy. That is released by the accumulation of warm temperatures. That's essentially a check to make sure spring has truly arrived. Now, I should mention at this juncture, while it's good to avoid frost damage, there's also, a negative aspect to waiting too long to initiate growth. Poplars especially, they're a shade-intolerant tree. Their strategy is to outgrow everyone around them in these disturbance habitats. So if they flush too late in the summer, they can't really fill that ecological role. So that kind of leads me into my main questions for this talk. First of all, I'm wondering broadly, as climates change, will we see changes in spring phenology in trees, specifically in bud flush timing? And if there are changes, will we see an increase in the prevalence of these frost damage events? Or on the flip side, might we see trees missing out on potential longer growing seasons by not flushing early enough? Or it could be that the plasticity of this trait is of the nature that trees are able to track changes in the growing season. Now, in that top left figure, the map of the Northern Hemisphere, I'm showing you data from a really good climate paper on showing change in the number of frost days over the past few decades. And the red areas, it's kind of hard to see, sorry, but the red areas have seen a decrease in the number of frost days, and the blue areas have seen an increase in the number of frost days. So broadly, it seems like the answer to my first question here is probably going to vary across space or vary with environmental context. Down here, in this kind of busy plot, I'm showing you a bunch of reaction norms. And so comparisons of bud flush timing for the same genotypes in different environments. And so one thing you can notice is that in a given environment, we often see a great genetic diversity for bud flush timing and applying some sort of genetic component to this trait. We see in some cases parallel reaction norms where the genetic rank order of genotypes is maintained across environments, but we also see plenty of instances of non-parallel reaction norms. So that's a hallmark of a genotype by environment interaction. So my second question here is kind of what is the genetic architecture of bud flush timing? In other words, what are the relative contributions of genetics, environment, and their interaction to the timing of bud flush? Finally, I'm wondering, if we can observe enough genotypes in enough environments, can we predict bud flush? Can we build models, predictive models that help us understand when bud flush might be happening for a particular individual in a particular environment? And ideally, we would move from kind of these discrete reaction norms comparing discrete environments to continuous reaction norms across continuous environmental space. If we can develop models like that, that would actually kind of help me answer that first question I've posed here. So I have a really cool study system I get to answer these questions with. I studied 2 poplar trees, Populus balsamifera, balsam poplar, and Populus trichocarpa, black cottonwood. You can see the range of balsamifera in blue and the range of trichocarpa in red. Now, these are 2 recently diverged species within the Populus genus, and they are still interfertile. So it's kind of hard to see, but there are little zones of range overlap kind of in the Pacific Northwest, Alaska, and Western Canada. It's well known that these species rampantly hybridize in those zones of range overlap. Now, I'm not going to talk too much about the hybridization story. Would love to talk about it after, but I will say here, I think for long-lived organisms like trees, natural hybrid zones are an amazing resource if you're interested in the genetic basis of a trait, because we see novel combinations of genotypes. It's basically nature making crosses for us that we wouldn't be able to do because of the long life cycle of these trees. However, you can't take advantage of natural hybrid zones like this if you don't sample them. So in 2020, my collaborators on this project went out and sampled 575 poplar trees in 6 distinct transects running through these hybrid zones. We sequenced the whole genome of all 575 of these individuals, so the color of the points in that left-hand map corresponds to % Populus trichocarpa ancestry based on admixture analysis of that genomic data. And for the rest of my talk, color indicates % Populus trichocarpa ancestry. One of the really cool things about poplar is it's unlike a lot of trees in the sense that we can clonally propagate it. So we took advantage of that and clonally propagated all 575 of these trees in multiple replicates to 3 common gardens, one in North Dakota, one in Virginia, and one down in Vermont just across the border from here. We also chose a subset of 50 genotypes, kind of trying to encapsulate a range of geography and ancestry. You can see where those genotypes came from. That subset of 50 we planted in 18 what we're calling mini gardens, so smaller common gardens. You can see their locations on the map there. And so you can see we kind of tried to cover a bit more of a diverse set of environments with these mini gardens. And I'm calling this my validation data set because it will come up later for validation. But before I get into that, I just want to show you what the data looks like from the bigger common gardens I was talking about 2 slides ago. So this plot is pretty busy. We can circle back to it if we have time, but basically what's going on here is each panel is a different common garden in a different year. So that top row is 3 years in the Virginia common garden, for example. On the y-axis, I'm showing you cumulative growing degree days to bud flush. So that's essentially how many days did it take for a tree before it flushed its bud, where each day is weighted by how warm it is. So we're kind of trying to encapsulate a more biologically meaningful way of measuring bud flush timing, because like I told you, eco-dormancy is broken by that accumulation of warming. It's not necessarily based on calendar day. On the x-axis, I'm showing you information on these trees' home environment. So points on the far right side of the x-axis are from cold environments where winter is very long. Trees on the far left of the x-axis are from warmer environments where winter is shorter. And so when we plot the data like this, what falls out are these kind of weak but significant quadratic clines, where the earliest individuals to flush their bud are from from colder environments and are generally of Populus balsamifera ancestry. Meanwhile, the individuals that need the most warming, the most number of cumulative growing degree days before flushing are from intermediate environments, which is really interesting. The last thing I just want to point out on this plot is the very, very strong environmental effects we see. So like some of the latest to flush individuals in Vermont in the middle would have been some of the earliest in Virginia, which is interesting. But how do I use this sort of data to answer the questions I posed at the beginning of my talk? Well, methodologically, I've been using genomic prediction. So for those of you who aren't familiar with genomic prediction, it's basically operating on the quantitative genetic wisdom that variation for a given trait is just the sum of variation due to genetics, variation due to environment, and their interaction, or G by E, genotype by environment interaction. So I incorporate genetics into these models using a genomic relatedness matrix, and I actually build two of those, one from SNP data and one from structural variant data. I can talk more about that at the end if people have questions, and I will compare the two in one slide from now. Similarly, I include environmental data in these models using an environmental relatedness matrix. And that relatedness is based on a suite of environmental variables I've listed on the slide. Almost all of them are related to the depth or duration of winter cold, save for photoperiod, which I include in there, as we think photoperiod has a slight effect on spring phenology in poplar. So now I'm going to dive right in to some of the comparisons of different genomic prediction models I've fit. So I'm showing you three different models here. The top panel is how the model performed in cross-validation. So model accuracy is just the correlation between predicted and observed bud flush timing. The bottom panel is the variance components of each model. So if we start on the far left, the first model I'm showing you there only includes genetics. It was worth a shot, but turns out you cannot predict bud flush timing very accurately from genetics alone. And you can see our predictive accuracy is quite low, and a great deal of variance ends up in the residual. We move to the middle model, and now we're including an effect of environment and an interaction between genetics and environment, and we do much better at prediction. The only difference between the middle model and the one on the far right is on the far right, we've partitioned G by E into an interaction with discrete environment and an interaction with continuous environmental variables that are in that environmental relatedness matrix I was talking about. I'm kind of bummed with this result. I think the difference in those two segments suggests that I'm missing some sort of latent variable that's not making it into the environmental related matrix. So if anyone has any thoughts on that, I'd love to talk after, but suffice it to say, when we include environment and an interaction between environment and genetics in these models, we do quite well at predicting bud flush timing, at least in random cross-validation. The last takeaway from this on the top, you'll notice I have two distributions for cross-validation. The blue is for when I use SNP data as the genetic component of the model. The yellow, or goldenrod, I guess that is when I use structural variant data. And it's just an interesting thing to note. We do slightly better when we use the structural variants. So I thought that was cool. However, I don't think that random cross-validation is really the best way to test these models. So the next few slides, I'm going to talk about some of the more robust tests of the model that I did. So the first is trying to simulate a situation where we're predicting bud flush for novel genotypes, or genotypes that the model hasn't seen before. So remember, my data all comes from 6 distinct transects, and I'm showing you the 3-number code that we've named these transects with. And so what I did to simulate unseen genotypes or unobserved genotypes for the model is I would hold back all of the data from one transect at a time, and then again compare the observed bud flush timing to what the model predicted. And as you can see, pretty much across the board, we do pretty well predicting for unseen genotypes. So the next thing I wanted to test was unobserved or unseen environments. So this is where those mini gardens in my validation data set come in. And as you can see, in certain environmental contexts, prediction accuracy absolutely plummets. And so that's generally in unfamiliar environments. And when I say unfamiliar, I mean environments that are unfamiliar to the model in the sense that they're environmentally distinct from the bigger gardens that we use to train the models, but also environments that are unfamiliar to the poplars. You can see we do the worst at predicting in Texas and California, And those are just environmentally very, very strange for the poplars, very far from the types of environments that they're used to. So perhaps, our prediction accuracy is just better when the poplars are growing better. You can see kind of in these areas that are closer to the poplar range, our prediction accuracy goes up. We do really well predicting in Virginia as well, but that's because we also had one of the bigger gardens there that was used to train the model. So, with the caveat that we don't do too well predicting into novel environments, I will now present data predicting into novel environments, specifically the future. So what I did here is I got projections, pooled climate projections for a couple business as usual scenarios for the year 2080. And what I did is I did two things with that data. One is I got those projections for every single sampling location that we originally got these trees from, and I used my model to predict bud flush timing at each of those sampling locations, so each tree's home environment for the year 2080. I also used that data to try to find the projected day of final frost for 2080, and that's what I'm plotting against each other in that left-hand plot. So a point on the one-to-one line, that would be a tree that's predicted to flush its bud on the very day that winter ends, if that makes sense. So you'll notice most of the points are above the line. That's implying that in those trees' home environments, the growing season is likely to get a bit longer, but they will struggle to keep up with, or they'll struggle to track that earlier growing season. There'll be plenty of growing degree days that pass before they finally flush their bud. However, there are a couple points below the line, and so those are the trees that we're worried about flushing too early and then getting hit by a return to winter temperature. And so I've also plotted the difference between these two axes on the map there, and so the dark orange points are the ones that we're worried are going to delay bud flush too long, and the lighter blue points are the ones we're worried about frost damage for. Now I just have one last slide before I wrap up here. I just want to wave my hands really fast about some actual specific genetic markers involved in this. So the models that I've been presenting on for this talk, like I said, genetics makes it into the model in the form of genomic relatedness matrix. But like I said, I'm also interested in the effect of specific markers. And so I have been fitting genomic prediction models that do model marker-specific effects. I just wanted to mention these two outliers here. They're interesting to me not only because of their effect size, but also because the genes that these structural variants neighbor have been shown in trees and other plants to affect phenology. So that's pretty validating for me. So I think I've answered all of the questions that I posed at the beginning of the talk now. And with that, I would happily answer any of your questions. Thank you so much.
Hi, everyone. Thanks for coming out today. I'm excited to tell you some new results from probably the newest type of research going on in my lab, asking about new phenotypes that might be associated with how parasites are adapting to their hosts. Before I get too far into this, I want to acknowledge the people who did most of the work. So this project was started by my master's student, Bryce Solon, who recently finished, an Alaskan native, as you can tell up here, very excited about these fish and these worms. Lily Wagner was a technician in the lab, and Javi Benavente did some of the genetics that I'm going to talk about. So evolution conference, we're all interested in phenotypic diversity that's out in nature, and sometimes when we look out in nature, it's obvious. That might not be easy to study, but when you go to the Galapagos and you look at beaks of finches, you can say on this island there are different beaks, and they probably do something different. That's great. Sometimes but how do we want to classify this kind of variation? Oftentimes we use the term functional trait because we suppose that there's some kind of specific function that is different between these different sizes of beaks. And that's really important for telling this adaptive story. And sometimes it's not easy enough to just go into one space at one time point and find this kind of variation. In the case of stickleback, which is what I study, sometimes you actually have to look across lots of different spatial variants and over time, but then these functional variants pop up. You're able to identify and tell adaptive stories about what's happening with the kinds of traits that we see. But what if you don't see any traits? We study this tapeworm, which essentially, I love the tapeworm, but it's a blob, right? You're not going to see a whole lot of functional traits just popping out of this when you start looking over space. So how do we start to explore these kinds of other organisms which might not have as obvious types of phenotypic variation? And I'm not the first person to ask this question. There's a lot of parasitologists that are encouraging us to start thinking more about functional traits in parasites. There's a couple nice papers, including this one I list up here, where they list a bunch of different categories if you're interested in parasites, where you should start measuring different phenotypes that are pretty easily accessible to ask how those things might be connected to ways and adaptive functions. And just to give you some examples of these things, like classic ecological trends that we might not think about in parasites absolutely do hold. Latitudinal diversity gradients, where you go from see differences in the size of the parasites, differences in their niche breadth, the number of hosts that they might be occupying, and even in their geographical ranges. Like, these are all things that we should start thinking about, I think, a little bit more when we start thinking about parasites. But when we start asking about variation over space, we then have to ask about the mechanisms that are associated with explaining that variation. Are they plastic differences across space? Are they heritable? Or are they some kind of interaction between these two things? So a very brief introduction to the system. If you've been to any other stickleback talks, you might have heard about some of this stuff before. The tapeworm that we study in our lab is Schistocephalus solidus. It's an amazing model system for a lot of different reasons. Has a complex life history, but the important points for today are that its obligate host is a three-spined stickleback fish. It must go through that fish at some points in its life in order for it to persist and then get into a bird. It can infect lots of birds, lots of copepods, but it must go through a three-spined stickleback. That puts a tremendous selection pressure on that worm to get into that fish. There's a tremendous selection pressure on the fish also when you have all of these worms coming out of a single fish, selecting that there's really coevolutionary dynamics here on the two species interacting together across their range. And the really exciting part about this, at least I think in terms of the phenotypic plasticity session that we're in, is that we can replicate these infections in the lab. So we can do common gardens of worms from different areas and disentangle their heritable versus their plastic effects. So we've done a lot of work on stickleback now, looking across lots of different types of populations, and we've put together a pretty neat story about the history of the host evolution. And it's important for the plasticity I'm going to tell you about in the parasite, so stick with me here for 1 second. So the history of this, and what we have here is a graph of 56 populations of stickleback that Dan Bolnick collected on Vancouver Island, and then looking at the infection prevalence across different regions, different populations. So the first thing that we were all able to put together, this is work of Natalie Stelnell, also in the back, is that basically marine fish lack infections, but it's because they're cestode naive. The worms can't live in marine environments, so they never get exposed. But as soon as we expose these guys to worms in the lab, they get infected at very high levels. So you start out with a naive host. But then these naive marine hosts move into fresh water where they get exposed to the cestodes. And then they evolve resistance in lots of different ways across lots of different environments. I can't go into all the forms of resistance today, but they include things like suppressing how the worms grow after infection, lots of differences in innate immunity, which controls the parasite infections, and lots of other things that control parasite growth or other aspects of its life, including fibrosis, peritoneal fibrosis, which is actually really bad for the fish. And interestingly, we have some evidence that some of these traits are actually can be quite plastic. Depending on the environment in which you raise the host, you have different types of immune responses which have different consequences on these other traits that we're interested in. So there might even be some plasticity at the level of the fish there. And we even found some really cool evolution of tolerance in some of these populations, which I just want to tell people about. If you haven't heard about it, it's kind of fun. We have the whole spectrum here. But what about the parasite? So stickleback, yeah, we look across lots of different populations. We can ask lots of different things and see what's happening in terms of the adaptive radiation of the fish. Can we do the same thing with the parasite? Yes, we can. So basically, this is the same story I just told you, where we're now asking about transitions from marine to freshwater, but I just want to give you a macro geographic picture of what happens with the parasite. So starting with these marine fish, we expose them to worms from two very different places, Canada and Norway, and these fish get infected at very high levels independent of parasite. We can then separate and ask about freshwater fish and expose them to their local parasites or foreign parasites and ask what happens with infection success. In these cases, we find that the worms get in when it's local worms, but they're at much lower levels of infection than we find in this ancestral marine host. This means that there's been host evolution that's gone on that's suppressing infection. But we can also show that the worms, when we expose these fish to foreign worms from different continents, almost none of them get in. So that suggests that the worms must have evolved some kind of counter-adaptation that's specific to their different freshwater hosts. So there's independent evolution, like different forms of resistance happening here and different counter-evolution that's going on. So this means that this is what leads us to believe that we do have a pretty cool coevolutionary system here. But at that macroevolutionary level, I mean, it's, there's differences in infection success here, but I want to get down to particular functional traits. And I can't always just go over and sample a lot of fish from Norway and be able to do the common garden with all these things. So that brings us to more of a microgeographic variation to ask, well, what do we know about worms across local landscapes that might inform how we think about plasticity? So the first thing I'll just show you, and this is just really quick data, we're still really trying to parse this out, but when we start looking at variation, genetic variation in the worm across the Pacific coast, so from Alaska down into Vancouver Island, we can ask, is there any genetic structure in this parasite that might allow or infer that local adaptation might be going on within this? So we've now got lots of worms from lots of years, from lots of populations, and the general pattern is this, is that yes, there is structure, that we do see that there is variation along the major axis here going from Alaska down to Vancouver Island. But within any of these populations, it's a mess. Basically, you have tons and tons of gene flow that's going on across the worm populations, very different from what we find in stickleback, which are restricted to their watersheds. And so that means there's just tons of mixing going on. And why is that? Well, the terminal host for these worms is a bird. So they're getting around a lot. So this might actually make it very hard for the worms to have any local adaptation to a specific kind of fish host. Maybe they have to be prepared for getting dropped into lots of types of fish hosts, which have different types of immunity. So I also don't have a lot of time to talk about the genetics, but I am excited to think about some of the population level things, actually kind of similar to the first talk in talking about population differences depending on where you sample individuals, but even within a single lake, we actually find huge differences in inbreeding coefficients dependent upon year within these tapeworms, which are hermaphrodites. And so I think this also has a huge effect on the kinds of phenotypic variation that we're going to see popping out within populations. So this kind of a graph just shows that what kind of, like an F statistic of 0 means that basically expected number of heterozygotes that are popping out of a population. But when we look across a few different other populations in different years, hopefully this will pop up here, we actually find huge differences in trends where we can have shifts going to have populations becoming much more inbred over time in the same lake as opposed to other ones that become much more outbred over time. And this is over the course of like 5 or 6 years. So huge differences here in genetics that we just don't know how to deal with yet. All right, finally, phenotypes. I keep promising that there's like a phenotypic plasticity. What can we say about parasites from different populations and whether or not they have some kind of plasticity in particular phenotypes? So what Bryce, my student, did was he went out to 6 different areas of Alaska and sampled worms from lakes that the important part of the story is whether or not they had fish that were long-term freshwater. So these guys over here that had a long chance to evolve some kind of resistance to the tapeworms, as opposed to other ones which are not marine, but where fish have recently colonized freshwater and have not yet had a chance to really adapt much immunity. So do we see differences between worms that come out of these two kind of ends of the spectrum? What he did was histology on the worms, really simple stuff. We didn't know what to look for, like again, blob, but like if you then stain the blob, maybe you'll find some interesting things that are coming out. So we did longitudinal sections and basically looked like at thin sections right here across the middle and just asking, what do we see in terms of the anatomy of these worms? And they're actually really beautiful. There's a lot going on here. Bryce measured tons of things. We've got differences in muscle connectivity. We have lots of reproductive tissues. But the one that we really, really focused on is tegument. So this is the key innovation. People argue for platyhelminths, where basically they took their gut and put it on the outside of their body. And so this is now how they interact with the host. It's how they secrete molecules to manipulate the host, and it's how how they also absorb resources from the host. So it's this thin syncytium that is just absolutely amazing and a topic of a lot of research for developmental biologists because it takes a huge amount of energy to maintain this syncytium. There's a lot of little mitochondria staining right here, which is actually used to basically maintain this tissue. So what Bryce did, he went in and measured a lot of worms from a lot of populations, and particularly looked at controlling for size, which is the diameter of each worm, which controls like the age, and we also controlled for mass. And then we also went in and looked at the proglotid width. We looked at the tegument width on each one of these sections, so 3 sections per worm. All right, so all of that lead up, like what did we actually find? We find a huge difference in tegument width. So when we look at these long-term freshwater populations with resistant hosts, we find that the worms have like much larger teguments than the ones that are coming out of the recently colonized populations. And so this is just showing an allometric relationship with diameter. We have a few other covariates that we can throw in and like make it a little bit cleaner, but basically this explains 80% of the variation in tegument width when we fit our models. But we have lab infections also. So can we ask, are these heritable differences or are these plastic differences? And just very quickly to go through our expectations that if it's really the worm that's controlling tegument size, then if it's independent of the fish that it's in, that suggests it's parasite genetics that's driving it. If the host predicts the type of tegument that we see, then it suggests it's some kind of host genetics. Or maybe it's some kind of environmental difference that's popping out. And again, a very obvious result, but so I didn't put any statistics on this yet, but it's a huge interaction. When you go from the wild, where you find huge differences in tegument width, and then take those populations and expose them to all possible combinations in the lab, we lose the tegument width variation. This actually fits really well with previous work suggesting that there really is some kind of important training of the immune system that goes on in wild environments. And there's a genotype by environment interaction where these fish that are resistant need to be having their immune systems trained, and then that is somehow feeding back into the tegument that we see. So sometimes you have to use a microscope to see things that are actually really obvious, so I guess that's the kind of thing that we're finding here. But we're really excited about asking more about the adaptive nature of what's happening with this tegument. Can we even take one worm with a one-size tegument and surgically implant it into a different kind of fish and ask if there are actually fitness consequences for those animals? And really, we're just scratching the surface of the kinds of things that we're really interested in asking about what's happening in the adaptive potential radiation of this tapeworm with the fish that they infect. And with that, I think I might have one question, time for one question. None? Oh, yeah. Emily.
Do you see any difference in fish that have many tapeworms versus ones? Could it be the later tapeworms that come in and have a different phenotype?
Yeah, so we've controlled in the models for the number of worms per fish, and it's not a significant predictor of the tegument variation. Although you could certainly imagine that they get exposed, they mount a strong immune response, and then maybe the later tapeworms have a harder time. We just haven't seen that yet, but we could look more deeply. Yeah, Dan?
So you set up this contrast of what's the relative effect size of host genotype or parasite genotype in the lab experiment. What were those relative effect sizes? Either this did or it didn't.
Oh, I just didn't put it up. I mean, they're small. There is still a significant effect of host, but not of parasite. Yep.
Hello, everyone. My name is Stephen Dreyer. I'm a PhD candidate in the Ragsdale Lab at Indiana University. My research centers on developmental plasticity, and specifically, I'm working to identify the genetic architecture of a plastic developmental response that harbors natural variation across populations in the wild. So, explaining the nature and causes of phenotypic variation is a continuing challenge for evolutionary biologists. And this challenge is particularly highlighted in traits whose development depends largely on environmental conditions. An extreme example of such traits is polyphenism, the taxonomically widespread ability of a single genotype to produce environmentally specific, discrete phenotypes or morphs. And polyphenism results from a threshold response that is dependent on the amount of a specific environmental cue. And intermediates are typically rare, and this implies a switch-like mechanism. And this is a really useful tool for studying plasticity due to its binary output. For example, these pea aphids shown on the lower left typically do not have wings. They live on cactus. They eat the fruit of cactus. But when they experience low nutritional or overcrowding cues early in their life history, they will develop wings, allowing them to search for new resources. So a good model for studying the genetics of polyphenism is shark-tooth nematodes. And nematodes are really cool because they're super cheap to maintain and culture. They're these little tiny animals. They live on E. coli. They have a very fast life cycle, 4-day generation time before they are reproducing the next generation. And I work with this species, Pristioncus pacificus, which develops into either a non-predatory ST form, which is strictly microbivorous, But if it experiences environmental cues such as low nutritional availability or overcrowding, it can develop into this predatory EU form that develops a novel hook-like tooth that allows it to predate on other nematodes. This species is really cool because it's mostly hermaphroditic. It produces males very rarely, and it typically self-fertilizes, so it's really good for crossing experiments. And it is also homozygous across its genome for all alleles, so we can control for the genetic background. So we know a lot of the interactions that are occurring at the molecular level, and a lot of genes have been identified that are involved in this regulatory network. However, most of these genes have been identified in a single isolate in the lab. And because these isolates are homozygous, we really don't know the evolutionary history of these genes. We don't know how these networks are evolving. Are these genes under the target of evolution? I do want to bring your attention to the UD1 gene that is circled in red. That is the major switch gene that when we knock it out, we produce constitutive non-predatory ST forms, and when it is overexpressed, we produce constitutive predatory forms. But if I want to know how this polyphenism network is evolving, then I need to look at populations across the wild rather than a single strain in the lab. And so we know there's a lot of variation in the environmental response for this particular phenotype. And the phenotype that we look at for this polyphenism is its morph ratio production under different environmental conditions. And so we can see here on the x-axis, a lot of different isolates have been recovered from different locations. And on the x-axis, we show the percent predatory or EU form. And we can see across these isolates, there's a lot of differences ranging from near loss of the predatory form to total fixation of the predatory form. And I want to know what is the genetic architecture underlying this variation. So theory suggests that a regulatory network for a plastic response should develop in a stepwise manner. But it's still unclear whether a few large effect loci are involved or if this is occurring due to the interactions of many alleles. And so the way that I want to go about this is by using these two lines, Washington designated by blue and California designated by yellow. So these two isolates exhibit similar phenotypes under standard lab conditions. And this is often overlooked, but a classic prediction of quantitative traits is that populations are expected to harbor variation even when they produce the same ultimate phenotype. And so I wanted to know if there were similar mechanisms producing these phenotypes, but if there is variation, then maybe I can leverage that to understand how this is evolving under the hood, so to speak. So one way that we can do this is through the creation of recombinant and bred lines. Remember, these are self-fertilizing populations, so they're homozygous across their genomes. So indicated by red or blue and yellow, the Washington and California lines, I cross them together. The F1 hybrids are completely heterozygous. At the F2 generation, independent recombination events are going to ensure different combinations of the genetic background, and then I will self them for another 10 generations to ensure homozygosity across the genome. So I did that, and then I wanted to look at the phenotypes of these different backgrounds. So I'm showing the two parental lines in orange on the left of this graph, and so this figure is showing the percent morph ratio production under standard lab conditions. So I'm controlling for the environment. We're just strictly looking at the phenotype due to the genetic background. And you can see the parentals range from about 70% up to 100%. These are individual replicates, each dot. And I was able to create a continuous distribution of these morph ratios. So that's really cool because it shows that there is some sort of cryptic genetic variation that is being accumulated in these individual lines, even though they're producing the same phenotypes. And we're revealing this by reshuffling alleles into these different combinations. So my next step was to perform whole genome resequencing and obtain the genotype information for these lines. So I got the genotype information, phenotype information. I wanted to perform a quantitative trait analysis to associate these two things. So I did that. And it looks like chromosome 1 and chromosome X both show peaks of large effect contributing to this variation. So chromosome 1 looks like there may be some linkage across the left and right arms. I'm still trying to figure that one out, but chromosome X, importantly, has that UD1 major switch gene that I showed earlier, very important to the polyphenism. So I wanted to kind of take a look at the genetic backgrounds at these different loci. So Washington is still designated in blue, California is in yellow, and I'm showing those different backgrounds at the bottom of these graphs for each of the RILs. And importantly, the top predatory morph bias ratios have this California background at the chromosome X QTL. And in the interest of time, I'm just going to stick to the chromosome X. But this was really cool to see this, and it seems to be an epistatic interaction allowing the buildup of genetic variation. But there was another study that was published at the same time that I was working on this project, and they also identified this QTL region. And they found that there was transcription factor binding site differences, copy number variations for the UD1 switch gene. And I wanted to make sure we weren't finding the same thing and that I hadn't been scooped, hopefully. So I wanted to take a look at these two lines that I was using and see, could this be causal? So the California strain in yellow has 3 regulatory binding sites designated by stars. And it turns out that, yes, the Washington strain has fewer with just a single regulatory binding site. But that's not causal, just showing that there's differences. So I wanted to test this with CRISPR knockouts. So I'm showing on the left the number of regulatory binding sites, and on the right is the phenotype % predatory morph. So California, still very high. I generated 9 independent mutants, deleting either 1 or 2 of these copy numbers, and there was no significant effect on the phenotype. And so this is really cool because it's the same locus on this chromosome X, but it appears to be targeted by evolution independently multiple times. So this is not the same causal allele. And also importantly, none of the genes in this regulatory mechanism that we know of popped up under this QTL analysis. So that kind of indicates that we can find genes involved in networks in the lab populations, but that might not be how they're evolving in the wild. And so to further fine map this locus, I'm using CRISPR to target recombination events in order to switch genetic backgrounds rather than relying on natural recombination. So I've done 1 to completely exclude the UD1 gene, and there are no known genes within this blue region on the right. So I'm going to keep fine mapping this through CRISPR experiments. So in summary, a QTL approach has revealed hidden natural variation in the regulation of polyphenism. The genetic architecture shows a few QTL of large effect. I've identified a novel target of polyphenism evolution, and it seems that evolution can use disparate mechanisms to achieve the same phenotypic outcome. I want to acknowledge the Ragsdale Lab, everybody that has helped me out there. It's been really cool. Thank you. Any questions? Yes?
Are you looking at any other traits in your RILs that might be associated with some of the functions? Like you look at the morphological difference that defines the 2 different groups?
Yes. So
everything else that's adapted associated.
that is future work. I am looking at the magnitude of response to induction cues. Inducing one predatory morph or the non-predatory morph in RILs that already have these morph bias ratios. And then I will be doing a QTL analysis on that as well, seeing if we identify patterns with the loci that are involved with this mechanism. I'm also doing geometric morphometrics on the mouth form shape itself to see if there are any parts of the mouth form itself that are capable of evolving. Are there constraints on the evolution of the mouth form? Any other questions? All right, thank you.